When we talk about selecting patients in cancer medicine, I think we're talking about not only the clinical selection, but also a biomarker selection, right? Molecular oncology has made great impacts, but the greatest impacts, I think, have always been when we have a targeted agent in a targeted population. So selecting patients for systemic treatment in liver cancer has to take into consideration their performance status, their underlying liver function, and how well they will tolerate systemic treatment. We still do not have a biomarker for identifying patients who benefit most from the therapies we have, that being sorafenib or regorafenib. There's no clinical group that does not benefit. We do not say, well, you have this characteristic, so you should not be treated with sorafenib, assuming they're candidates for systemic treatment. That's changing in the sense that there are clinical studies that are ongoing that are looking at biomarker-selected patients that will, if successful, will be linked to a systemic therapy, a specific therapy. For example, tivantinib is being studied in a population of patients who have elevated expression of the c-Met receptor, hepatocyte growth factor receptor. There's a whole group of drugs in early development for patients who have alterations in the FGF19 and FGF receptor 4 axis. There's a large phase 3 study evaluating ramucirumab in the second-line setting, but only in that subset of patients who have an elevated alpha-fetoprotein. So as it stands today, liver cancer is not where breast cancer is, or lung cancer for that matter, with ALK and EGFR, and breast cancer HER2 or estrogen receptor. But we recognize that we need to get there, and those studies are ongoing.